Immunology Insights, Basics and Beyond, Chapter 50, Tuberculosis and Immunity. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. In our previous chapter, we explored how HIV cripples the immune system, leading to devastating opportunistic infections. One of the most important of these, and one of the humanity's oldest adversaries, is tuberculosis, or in short, TB. Today, we will dive into the remarkable relationship between Mycobacterium tuberculosis and the immune system, a complex dance of activation, evasion, and persistence that has shaped global health for centuries. So let's talk science. Let's start with the global impact of tuberculosis. Tuberculosis is caused by the bacterium Mycobacterium tuberculosis, or MTB, transmitted through airborne droplets when an infected person coughs or sneezes. Despite being treatable, TB remains a leading cause of deaths worldwide, infecting nearly a quarter of the global population latently and killing over 1.3 million people each year. While TB was nearly eradicated in high income countries, it remains a major problem in regions such as sub Saharan Africa, South and Southeast Asia, and parts of Eastern Europe, where poverty, malnutrition, overcrowding, and HIV co infection drive transmission. The bacterium's resilience, coupled with drug resistance, keeps it among the top infectious killers even today. Now, how does the infection begin? Well, when MTB enters the lungs, it is engulfed by alveolar macrophages, the very cells meant to destroy bacteria. But MTB is no ordinary pathogen. It can survive inside macrophages, resisting the acidic environment and blocking the fusion of phagosomes with lysosomes. This triggers an alarm. Macrophages release IL-12, TNF alpha, and chemokines, recruiting more immune cells, including dendritic cells, CD4 positive Th1 cells, and CD8 positive cytotoxic T cells. These cells surround infected macrophages, forming a structured immune fortress known as the granuloma, the hallmark of tuberculosis. Let's have a closer look at this immune battle. So, containment versus disease. Inside the granuloma, a delicate balance unfolds. Th1 cells release interferon gamma, activating macrophages to kill or contain MTB. Cytotoxic T cells and NK cells help eliminate heavily infected cells. And macrophages transform into epithelioids and multinucleated giant cells, walling off the infection. If the immune system succeeds, MTB enters a latent state, alive but dormant. About 90% of infected individuals remain in this latent phase, showing no symptoms. However, if the immunity wanes, for example due to HIV, malnutrition, or aging, granulomas can break down, releasing bacteria and causing active TB, characterized by chronic cough, fever, weight loss, and night sweats. In this stage, the bacteria spread through coughing, infecting others, often family members or close contacts. Let's see why the immune system struggles. MTB has evolved remarkable immune evasion tactics. First, intracellular survival. It prevents phagolysosome fusion and neutralizes reactive oxygen and nitrogen species. Second, antigen variation. It alters its surface lipids and proteins to confuse immune detection. Third, immune modulation. It skews cytokine responses, suppressing Th1 activity to avoid destruction. And fourth, latency. Dormant bacteria express minimal antigens, escaping immune recognition altogether. In essence, TB doesn't just hide, it manipulates the immune system into tolerating its presence. Now, what is the role of CD4 T cells and the HIV connection? The importance of CD4 positive T cells in TB control became tragically clear during the HIV pandemic. In HIV positive individuals, CD4 depletion removes the immune coordination necessary to maintain granulomas, allowing TB to reactivate. Indeed, TB is the leading cause of death among HIV-infected patients worldwide. So let's look at immune pathology and host damage. Paradoxically, much of TB's damage comes not directly from the bacteria, but from our own immune system's overzealous attempts to contain it. As granulomas mature, persistent inflammation leads to caseous necrosis, a cheese-like breakdown of tissue at their center. If the immune response becomes unbalanced, these necrotic granulomas rupture into airways, forming cavities filled with infectious bacteria. This destructive process is driven by excessive TNF alpha, matrix metalloproteinases, and reactive oxygen intermediates released by activated macrophages and neutrophils. The result: extensive lung tissue loss, bleeding, and progressive respiratory failure. In healthy individuals, immune responses are tightly regulated, containing bacteria without significant tissue destruction. In active TB, however, the immune balance tips, defense turns into collateral damage. Of course, there is treatment, but also drug resistance. Standard TB treatment uses a combination of antibiotics: isoniazid, rifampicin, pyrozinamide, and ethambutol for at least 6 months. These drugs target different bacterial pathways to prevent resistance. However, incomplete adherence has led to multi-drug resistant, so MDR-TB, and extensively drug-resistant XDR-TB strains, now found in over 100 countries. Treating resistant TB can take 18 months or longer, with more toxic and less effective regimens. But what about vaccines, the BCG and beyond? The BCG vaccine, so Bacillus calmette-guerin, developed in 1921, remains the only approved TB vaccine. It protects children against severe forms like TB meningitis, but provides inconsistent protection against adult pulmonary disease. New candidates are in development. Examples include M72AS01E, showing 50% protection in adults with latent TB, or VPM1002, a recombinant BCG strain designed for stronger immune activation, or mRNA-based vaccines, leveraging COVID-era advances to target MTB antigens more precisely. These aim to induce more durable Th1 and CD8 T-cell responses, crucial for long-term control. What is TB's relevance and what are future directions? Tuberculosis remains a biological paradox, an ancient foe that thrives despite immune recognition. It continues to shape global health priorities and serves as a model for studying chronic infection, immune regulation, and host pathogen co-evolution. Modern efforts focus on: first, developing next generation vaccines for both prevention and therapy; second, creating shorter, more tolerable drug regimens; third, exploring host directed therapies to modulate immunity without increasing inflammation; and fourth, integrating TB HIV care to tackle both epidemics together. If these strategies succeed, we may finally shift the balance from containment to eradication. To conclude, tuberculosis has walked alongside humanity for thousands of years, turning our immune defenses into its own survival strategy. Yet, the same immune system that it exploits also holds the key to its defeat. By understanding how MTB hijacks and inflames our immunity, we are learning to build smarter therapies, ones that heal without harm and prevent disease before it starts. In our next chapter, we will move from bacteria to viruses, exploring influenza and other viral infections, and how our immune system's adaptability equips it to battle pathogens that mutate faster than any other. Before closing, I have a small request. If you enjoyed my content and want to hear more, like and subscribe. It's a small step for you, but it helps me a lot. So, see you next time and thank you for listening.